Welcome back to Divan's Biology. In this video series on respiration in plants, we are discussing the mechanism of respiration in detail. The mechanism of respiration is mainly divided into 4 stages, of which the 3 stages have been discussed in detail. Today we will complete the 4th stage, in which ATP molecules will be formed and oxygen will be utilized. Till now what we have seen? Till now in all the stages glucose is completely oxidized, but neither oxygen is being utilized nor huge amount of ATPs are released. So these two things will be accomplished in stage 4, which is electron transport system, along with oxidative phosphorylation. This ETS is actually performed in the inner membrane of mitochondria. Let us summarize the first 3 stages before moving on to the 4th stage. In the first stage, which is known as glycolysis, glucose was partially oxidized to pyruvic acid. This process was done in cytosol of the cell. In the second stage, pyruvic acid was oxidized to acetyl coenzyme A. This was done in mitochondrial matrix. It means after the formation of pyruvic acid in glycolysis, this pyruvic acid got transferred from cytosol to mitochondrial matrix. In the third stage, there was a Krebs pathway or Krebs cycle, and that was also accomplished in mitochondrial matrix. In Krebs cycle, only 2 ATPs were formed. In glycolysis, also the net gain of ATP was 2. But we are expecting huge amount of ATPs to be released from one glucose molecule, from oxidation of one glucose molecule. Now let us revise the structure of mitochondria. Mitochondria is actually a double membranous structure. This is the outer membrane of mitochondria. Inner membrane is actually highly folded in mitochondria. These folds are known as cristae. And the electron transport system, which we are going to study today, this is performed in inner membrane of mitochondria. Inside the mitochondria, it's matrix, mitochondrial matrix, and in between these two, outer and inner membrane, the space is there which is known as intermembrane space. Let us zoom in to this inner membrane, because this ETS is performed in this inner membrane. The membrane of mitochondria is made up of phospholipid bilayer along with the protein complexes in it. There are 4 protein complexes. This is the inner membrane of mitochondria. This area is the matrix of the mitochondria, and this is the intermembrane space. In the first 3 stages, we saw that reduced compounds were formed, NADH and FADH2. These reduced compounds need to be reoxidized. If these compounds are not reoxidized, then the whole respiratory process will come to the halt. So for reoxidation of these reduced compounds, this electron transport system is performed. The reduced compounds are NADH and FADH2. Individual electron transport proteins are organized in 4 protein complexes. These 4 protein complexes are termed by Roman numerals: complex 1, complex 2, complex 3, complex 4. First, third and fourth complex pump the proton from matrix to intermembrane space, thus creating a proton gradient. So electron transport system has 2 main roles. First, it oxidizes the reduced compounds. Second, it creates a proton motive force by using the free energy that is being released while oxidation reduction reactions. Now, let us start with the first complex. The first complex is NADH dehydrogenase. The name of first complex is NADH dehydrogenase. Electron carriers in this first complex includes a tightly bound FMN. FMN means FMN, flavin mononucleotide, which is structurally similar to FAD. This first complex contains electron carriers which are iron sulfur centers and tightly bound FMN. In this complex, NADH is oxidized and these electrons are then transferred to ubiquinone. Along with it, 4 protons are pumped. 4 protons are pumped from matrix to intermembrane space. Why protons are pumped? To create a proton gradient. In the second complex, okay, let us see what is ubiquinone. Ubiquinone is actually a small lipid soluble electron and proton carrier. It lies in between the inner membrane. It can diffuse within the hydrophobic part of inner membrane. It serves to transfer electrons from dehydrogenases to either complex 3 or, sorry, this is complex 3, to either complex 3 or to AOX. What is AOX? That will be discussed later. Now let us move to complex number 2. Complex number 2 does not pump protons. Complex 2 is known as succinate dehydrogenase. This succinate in complex number 2, succinate is oxidized to fumarate. During the oxidation of succinate, the electrons are transferred to ubiquinone via FADH2 and iron sulfur centers. While studying about Krebs cycle, I told you that all the enzymes of Krebs cycles are soluble in matrix of the mitochondria, but only one enzyme is there which is not present in mitochondrial matrix. Instead, it is attached to the inner membrane of the mitochondria. That enzyme is succinate dehydrogenase. This enzyme oxidizes succinate that is being formed in Krebs cycle. Now let us move to third complex. Third complex is known as cytochrome BC1 complex. This third complex oxidizes the reduced ubiquinone and transfers the electrons via iron sulfur center to B-type cytochromes and cytochrome C1, finally to the cytochrome C. Now what is this cytochrome C? Cytochrome C is a small protein on the inner membrane of mitochondria towards intermembrane space. This cytochrome C serves as a mobile carrier, electron carrier between complex 3 and complex 4. In complex 3 also, 4 protons are pumped per electron pair. In complex 1, 4 protons were pumped from matrix to intermembrane space per electron pair, while in second complex, no protons are pumped. Now let us move to fourth complex. The fourth complex is cytochrome C oxidase. It's cytochrome C oxidase. This is the terminal oxidase. It contains 2 copper centers and cytochrome A and A3, and this brings out reduction of oxygen to water molecules. In this complex, only 2 protons are pumped from matrix to intermembrane space. Structurally and functionally, this ubiquinone is similar to the plastoquinone of photosynthetic electron transport system, and functionally and structurally, this complex 3 is similar to cytochrome B6F complex of photosynthetic electron transport system. Now plant mitochondria actually have 2 different pathways for oxidizing NADH. Why they need 2 different pathways? Already we have complex 1 in which NADH dehydrogenase oxidizes the NADH to NAD. Actually, electron flow through this first complex is sensitive to inhibition by few compounds like rotenone and pyrazidine A. Now what is a rotenone? Rotenone is a botanical pesticide that is naturally present in some plants. So in that case, the plant mitochondria have rotenone insensitive dehydrogenases, NDIN, this one. This is which is attached to the mitochondrial membrane, inner membrane on the matrix site, NDIN. This is rotenone insensitive dehydrogenase, NDIN and NDEX. This is on the inner side of the inner membrane. This is on the outer side of the inner membrane. Outer side means towards inner membrane, intermembrane space. So these two are this one and this one. These two are rotenone insensitive dehydrogenases. The electrons are then passed on to, after oxidation, the electrons are passed on to ubiquinone from both rotenone insensitive dehydrogenases. Plants also have one additional pathway for oxidation of ubiquinone and reduction of oxygen, and that pathway is alternative oxidase, AOX. This alternative oxidase is present on the inner membrane, on the inner side, that is matrix side of the membrane. The electrons from ubiquinone are passed on to AOX. This pathway is insensitive to inhibition by cyanide, carbon monoxide and a signal molecule nitric oxide. Till now, we have utilized oxygen to form water, but still we have not formed huge amount of ATPs. So for that, plants have complex 5, which is known as ATP synthase. Complex 5, ATP synthase. This complex will form huge amount of ATPs. Till now, in this electron transport system, we have seen that a proton gradient is formed. Per electron pair, 4 protons were pumped from matrix to intermembrane space from complex 1. 4 protons were pumped from complex 3 and 2 protons per electron pair were pumped from complex 4. So now proton motive force has been created. To make a balance, these protons will pump back to matrix, but they cannot pump back to matrix from any other channel except complex 5. This complex 5, this ATP synthase has 2 parts, FO and F1. This O, subscript O, is for oligomycin sensitive. FO is in the intermembrane space and F1 is towards the matrix side. These two are rotary motors and they pump back protons from intermembrane space to matrix, and in the meantime this F1 part will form ATP from ADP and inorganic phosphate. The amount of ATP formed depends on the substrate. If it's NADH, then 3 proton pairs will be pumped back from intermembrane space to matrix and 3 ATP molecules will be formed. So 1 NADH will form 3 ATP molecules. Similarly, 1 FADH2 will form 2 ATP molecules, as it will pump back 2 proton pairs from intermembrane space to matrix via ATP synthase. So each NADH will form 3 ATPs, each FADH2 will form 2 ATPs. In previous stages, we have seen that many NADH were formed and FADH2 was formed in Krebs cycle. So in this system, 1 NADH will form 3 ATP molecules and 1 FADH2 will form 2 ATP molecules. So finally, huge number of ATPs will be formed. The total energy currencies produced in this mechanism of respiration will be discussed in next video. I hope you are clear with the stage 4. If you have any doubt, you can write in the comment box. Your doubts will be clarified immediately. Thank you.